Good morning. My name is Ella Wilson, and today my thesis partner, Abigail Ng, and I will be presenting our thesis entitled Investigation of Biofouling Mitigation Arrangements Using Ultraviolet-C Light. Before I begin, I would like to acknowledge several figures who have been instrumental throughout this project. First, I would like to thank our principal advisor, Professor McMahon, for his invaluable feedback, guidance, and encouragement over the last year. I would like to acknowledge Professor Gulabeva, our reader, and Professor Harris, our editor, for ensuring that Abby and I are able to effectively communicate our findings. Next, I would like to thank Professor Martin and Mr. Doherty for their assistance in the electrical design and construction portions of our experiment. And finally, I would like to thank Professor Boyd, who first introduced us to the application of UV light as an anti-fouling approach. Biofouling is defined as the unwanted accumulation of marine growth on submerged surfaces, such as ships' hulls, water inlets, grates, and offshore structures. Marine growth may include microbiology in the form of biofilms, or macrofouling, with the adherence of organisms such as barnacles or seaweed. Fouling causes several functional and financial effects for vessel owners, operators, and the general marine environment. Fouling increases the roughness and frictional resistance of a ship's hull, reducing the operational efficiency of the vessel, increasing fuel costs, and causing excess carbon emissions. In Figure 1, the biofouling growth trends over time per organism type can be seen. In the Long Island Sound, a growth plateau is estimated to be reached at approximately 2 weeks of exposure time. There are several methods used in the industry today to address the issue of biofouling on ships' hulls and equipment. Copper biocidal coatings and copper anodes both operate under the same principle. Copper is released into the water surrounding the applied surface, poisoning anything and everything within the proximity. Not only does this kill the marine organisms responsible for biofouling, but anything that eats these organisms will also be affected, moving up the food chain. The largest issue from these types of coatings, however, is when copper settles at the bottom of the water column. The high copper concentration causes dangerous conditions for sessile organisms like the coral seen in Figure 2, whose proteins become damaged. As a result, the coral is never able to recover, even after copper is removed from the environment. Next is movement-driven coatings. These specific coatings are typically turned to because they do not contain any biocides. The physical surface of these coatings is smooth, so the biofouling fails to adhere, but this only works if the vessel is in motion. Finally, particularly in the application of sea chests and piping systems, chemical treatments are used. Trace amounts of these biocides are extremely harmful to the marine environment, as well as very expensive. Several tests in this realm of work have been done recently with biofouling and UVC LEDs. An LED is a light-emitting diode. The Netherlands Defense Science and Technology Group obtained results using UVC LEDs embedded in silicone in both seawater aquariums and open water testing. Electrical power was varied in the aquarium setup to determine the dependence of mitigation effects on supplied amperage. The same group then tested the same UVC LEDs with silicone in Melbourne, the North Sea, Northern Europe, and Singapore to determine the effectiveness of mitigation in various locations, weather conditions, temperatures, and salinities. The conclusions of these studies are that biofouling mitigation effectiveness is directly proportional to supplied power. Additionally, this mitigation method is effective across a wide range of conditions. The Australian Government Department of Defence experimented with UVC LED lattices in sea chests. This experiment demonstrated a limit of biofouling growth when considering the effective exposure. Upon the conclusion of the tests, it was discovered that the biofouling growth will encroach upon the LED, maintaining a constant distance once a certain amount of time has passed. This radius will not change, no matter how long the test is conducted for, demonstrating an asymptotic relationship. This brings me to the goal of our experiment. The objective of this thesis is to determine an optimal substrate to allow for even distribution of UVC light in order to mitigate biofouling at a level comparable to currently used biocidal coatings. The presentation of our project will proceed as follows. I will discuss the theory behind our approach and describe the experimental design process. At this point, I will pass the presentation to Abby to discuss the construction and testing procedure. Finally, Abby will go over our results and conclusions, as well as make recommendations for future work. So how does UVC light work to mitigate biofouling? Ultraviolet light or radiation is a form of energy produced by the sun. It is made up of photons traveling in wave-like patterns. This light is categorized on the electromagnetic spectrum by its respective wavelengths. The ultraviolet spectrum ranges from approximately 200 to 400 nanometers and falls between X-rays and visible light, as can be seen in Figure 3. UV light is typically divided into three categories: UVA, UVB, and UVC. UVA rays have the longest wavelengths, while UVC rays are the shortest. The long UVA and some middle wave UVB is generally transmitted through the atmosphere. The short wave UVC, or germicidal light, is the highest energy portion of UV light. It ranges from approximately 200 to 280 nanometers. When produced by the sun, it is generally absorbed by the Earth's ozone layer. However, it has been used in various marine and non-marine applications, such as the medical sterilization of air, water, or surfaces, and in the treatment of ballast water on board ships. In addition, relatively recent development of microtechnology has led to the increased commercial availability of UV LEDs, increasing the potential for their use in large-scale or industrial settings. UVC light has the ability to create DNA lesions. The four main building blocks of DNA are adenine, guanine, cytosine, and thymine. The chemical breakdown of the DNA base structures can be observed in Figure 4. Adenine and guanine are both purines, which means they have two nitrogen-containing rings that make up their main structure, as pictured in the red boxes. Cytosine and thymine are pyrimidines, which means they only have one nitrogen-containing ring in their structure. UVC light has the ability to cause pyrimidine dimers in the DNA structure. A pyrimidine dimer is an intrastrand crosslink in the DNA structure, where one pyrimidine will develop a covalent linkage with an adjacent pyrimidine. In this case, that would mean a pyrimidine dimer would form between cytosine and or thymine, which would be our representative pyrimidines in Figure 5. These can form whether the DNA is single-stranded or duplex. However, if the DNA is duplex, this mutation will only occur in a single strand of the DNA. UVC light can cause two types of these mutations. The first is the CPD, or cyclobutane pyrimidine dimer mutation, which is caused by two parallel covalent linkages, identified in Figure 5. The other type of mutation is the CPP, or pyrimidine pyrimidone photoproduct mutation, which is created with one cross-linkage. This is often referred to as the 6,4-PP mutation, as this is the most common type of CPP mutation. In both of these mitigation types, the overall DNA structure is altered. When this occurs, the DNA and RNA polymerases are unable to replicate the structure, which is vital for microbiology colonies to continue to grow. Because the microbiology colony simply stops expanding and it does not affect the organism's survival odds, no predators that prey on these organisms are affected, contrary to the copper biocidal coatings that tend to move up the food chain. For our experiment, UVC LEDs with a wavelength of 275 nanometers were selected and adhered to A36 steel plates, as this is a very common grade of steel used in shipbuilding. In previous work, silicone was used as a light distribution medium. However, to be commercially feasible, a marine-grade durable substrate must be used to distribute light that can withstand harsh conditions. Additionally, because biofouling coatings that operate on stationary vessels typically use biocides, this new task requires coatings that are not initially designed for anti-fouling purposes. 3 different marine-grade coatings were chosen to test as light distribution mediums: epoxy, polyurethane, and ceramic. These coatings were chosen because they are designed for and used in the marine industry frequently, but all offer differing physical and chemical properties that may affect their light distribution capabilities. A copper anti-fouling paint was also included in our trials as an industry standard baseline, in addition to bare steel plates that would allow us to observe unimpeded biofouling growth. In addition to these primary trials, a second set of coated plates were constructed to be a control group to test coating degradation from UVC exposure over a longer period of time. I will now pass the presentation off to Abby to discuss the construction of the experimental apparatus.
Thank you, Ella. To test the efficacy of the 3 marine coating types selected, several steel plates were coated and fitted with UV LED prior to continuous underwater exposure. Following the length of the testing window, the plates were retrieved for data collection and analysis. I will first discuss the materials and physical construction involved with the underwater components of our primary trial, with a focus on the setup of each plate. Each of the 10 underwater plates was built upon a 4x4-inch A36 steel base plate. A single 275 nanometer wavelength UVC LED diode was affixed at the center of each of 6 plates. 2 plates each were coated with JB Weld epoxy pro-grade, Samurai 2K polyurethane marine coating, G-Technique ceramic coat, and Blue Water copper shield bottom paint. The final 2 were left bare. A Danielson 24-inch fold-up crab trap was modified to house and suspend the plates over the length of the testing window. Figure 7 illustrates the coated plates suspended in the crab trap. The plates were arranged to limit interaction between different light sources. The white wire through the top of the trap connects to the powering supply, housed on the floating apparatus. A floating apparatus was designed to provide continual power to the underwater plates and may be seen in Figure 8. 2 panels of Dow Blueboard XPS, a form of rigid, closed-cell styrofoam, were used as a base of the floating components. The foam panels were 2 by 24 by 24 inches and were affixed to allow rotation. The powering arrangement consisted of 2 power sources: a 20-watt waterproof solar panel mounted to the left, and a 12-volt lead-acid backup battery housed in a watertight enclosure. A solar charge controller was used to direct power from the panel to either the underwater plates or to charge the backup battery. This allowed for continual powering of the plates day and night. In addition, voltage divider and potentiometer were utilized and housed in the watertight enclosure to ensure that the same power level was delivered to each underwater plate. The floating apparatus and underwater setup were connected by tie-off rope and underwater cable, with additional slack to minimize stresses. The primary trials were designed to submerge 2 of each plate type. The bare steel and copper biocidal painted plates were included as controls, in addition to the UVC lit arrangements with epoxy, polyurethane, and ceramic coatings. All plates were suspended and submerged in the apparatus described. Given the opportunity for practical marine growth in the Long Island Sound, the experiment was conducted at the Webb Beach with a design test window of 2 weeks of underwater exposure. This duration was selected to align with a plateau in marine growth. The test was truncated, however, to approximately 1 week due to harsh weather and was conducted between March 10th and March 18th. The testing depth varied between approximately 3 and 12 feet, and a 45-pound weight was used to anchor the underwater setup and minimize any movement. Figure 9 shows a view of the Long Island Sound, with the red circle indicating the visible floating setup as seen from the Webb Beach. In addition to our primary trials, a series of 6 shoreside control plates were set up for continuous monitoring of UV light and coating degradation. These included 2 plates of each epoxy, polyurethane, and ceramic coating arrangements. 1 plate of each coating type was submerged, while the other was left in open air. The open air set may be seen in Figure 10. You may note the tape on the rightmost plate. During the implementation of the primary and control trials, it became apparent that ceramic posed several challenges to construction. Specifically, the ceramic coating was unable to produce a flush surface and proved too delicate to support the electrical components. Due to these challenges, ceramic was found to be unsuitable for our experiment, and it is not fully examined in our results. Controls testing indicated no significant degradation of coating quality over a 3-week testing period. There are 16 data collection points for each test plate previously described. In Figure 11, the UVC diode may be seen at the center of each plate, with each data collection point marked with an X. As previously discussed, the plates are 4 by 4 inches, so data was collected at distances of increasing 1/2 inch intervals. At each of these radii intervals, 4 data points were checked for fouling growth. This amounted to a total of 16 points per plate. Immediately following the retrieval of the primary trial plates, microbiology data was collected. At each X point, such as the one marked by the red box, a 1/4 by 1/4 inch area was scraped with a metal scalpel and then examined under the microscope at 40 times magnification. During the examination process, microscope footage was examined and images of microbiology growth on the plates were able to be observed at various data points. The various types, quantities, and locations of the growth were recorded. Figure 12 shows a still image of the Leptocylindrus danicus, a common diatom of the Long Island Sound, which was observed 1/2 inch away from a UVC LED on an epoxy-coated plate. In the examination of all test plates, 5 separate organisms were able to be identified, including several diatoms. For display purposes, we have chosen to use professional microscopic images, seen in Figures 13A through E. A diatom is a unicellular algae organism with transparent silica cell walls. Diatoms may be found in lakes, oceans, and waterways, and may occur either in solitary cells or in colonies. These are generally centric in a rounded shape with radial symmetry, or pennant in a cigar shape with bilateral symmetry. Each type of microbiology we observed was identified as a common organism found during the winter, based on existing data regarding phytoplankton in the Long Island Sound. In Figure A, you can see the Thalassiosira minima, which was by far the most frequent diatom observed. In Figure B, a clearer image of the previously described Leptocylindrus danicus can be seen, another diatom. Figure C shows the Thalassionema nichioids. We mostly observe these diatoms alone, but in larger numbers, they're known to link together to form chains. Striatella unipunctata, seen in Figure D, is another example of an algae diatom. Finally, in Figure E, you can see Thiothrix bacteria. This was the least frequently witnessed of all microbiology and was only found on the ceramic-coated plates. The growth types observed were in line with the previously discussed biofouling trends, with bacteria and diatom growth occurring over the truncated 1-week window. A longer testing window would allow for more varied fouling to occur. Above, the red dots denote microbiology identified on the test plates, recorded at their respective locations. On the left, you can see the average results of the 2 bare plates. In the middle are the epoxy plate. Finally, the right shows the polyurethane-coated plate. A total count of the average microbiology on each coating type may be seen above. On average, between the 2 primary trial plate sets, 61 microorganisms were seen on the polyurethane plate, 20 on the epoxy plate, and 30 on the bare plate. No growth was recognized on the conventionally painted control over the length of the testing window. The average quantity of microbiology observed on the bare plates is shown in figure 17 above, sorted by type. The x-axis shows the distance from the center of the plate, ranging from 1/2 inch to 2 inches away. The y-axis shows the count of microbiology averaged over the 2 identical test plates. 4 types of microbiology were observed on the bare plates. The most common diatom found on the bare plates was Thalassiosira minima, seen an average of 19 times per plate. The bare plates provides a baseline of growth. As the bare plates were neither coated nor lit, the distance from the center is of little note in this case. The microbiology observed on the epoxy plates is shown in figure 18 above. In this case, the distances along the x-axis indicate the distance from the UVC diode, as well as the center of the plate. 4 types of microbiology were observed on the epoxy plates. The most common diatom found was the Thalassionema nichioides, seen an average of 9 times per plate. No significant reduction in biofouling growth related to the distance from the UVC diode was recognized. However, the total biofouling level was less than that seen on the bare plate. The microbiology observed on the polyurethane plates are shown in figure 19. 2 types of microbiology were observed on the polyurethane plates, Thalassiosira minima and Thalassionema nichioides. Thalassiosira minima, shown in blue, was extremely abundant on one of our 2 polyurethane plates. Note that the y-axis is adjusted to accommodate this concentration. A clear reduction in growth towards the center of the plate can be seen, as expected. No other microbiology types were recognized for this plate, performing better than the bare or epoxy tests by variety. However, the count of Thalassiosira minima far exceeds that seen in any other test case. The summary table above displays the amount of growth on the epoxy, polyurethane, and bare plates, regardless of their proximity to the light. The amount is demonstrated by the count of each form of microbiology, where 0 indicates no growth. It can be seen from this data that epoxy showed the lowest amount of growth overall. Factors to consider while evaluating the trends of this data include the limited testing window, potential interactions due to the proximity of copper-based biocidal paint, and data collection methodology. As only limited points could be reasonably tested in this fashion, data can be more strongly relied on to provide an indication of growth types than serve as representative samples of the total population. Previous work has demonstrated that microbiology types may be immune to UVC light application. This may explain the high concentration of Thalassiosira minima seen on the polyurethane plates. In conclusion, it is clear that the anti-fouling potential of UVC light is not independent of coating type, and several changes in the growth were observed. The G-Technique ceramic coating selected was not a suitable substrate for UVC light anti-fouling applications. Epoxy and polyurethane remained potential avenues and showed advantages in the types and quantities of growth observed. Epoxy demonstrated a uniform decrease in microbiology levels observed, while polyurethane demonstrated a decrease in microbiology as a function of the distance from the UVC light diode. While no currently investigated UVC LED and marine grade coating biofouling mitigation arrangement was as effective as the copper-based biocidal paint, epoxy demonstrated the best capability for uniform light distribution. We'll now make recommendations for future work. First, the testing apparatus may be refined. We suggest designing for a longer testing duration than the minimum recommended 2 weeks, and for harsher weather conditions. The electrical design may be expanded to allow for remote monitoring or controls and further light cycling experiments to further research the ability of microbiology to recover. In addition, an investigation into copper-based biocidal leaching rates and repeated tests with isolated controls could help remove confounding factors. Finally, with an improved testing setup, we recommend similar tests in differing weather conditions to examine seasonal effects and changes in microbiology. Thank you for attending the presentation of our thesis, Investigation of Biofouling Mitigation Arrangements using Ultraviolet-C light. I ask you to please hold your questions until the conclusion of Sydney's presentation.